Hi class, we're going to look at a tutorial on identifying bacteria as the unknown identification. So I created this chart here, flow chart. You can work through it to see what your unknown is. So we are working with 841 Bravo. You may get another one shortly, but 841 Bravo is your first primary bacteria. Think of it as a patient's specimen, and you have to diagnose what the bacteria name is and make sure that the doctor is going to treat it properly. Okay, so the gram stain is the first one you look at, and if it's a gram positive versus a gram negative, you go each side. So our 841, for example, was a gram positive, and so then we go to this side, the right side. And then we look at the cell shape, and in our 841 so far, it's a rod shape, so that means it's a bacilli. And so then we'll go to page 77, gram-positive rods. That's kind of generally where we end up. If it was a gram-positive cocci, we'd go to page 74. If it was a gram-negative cocci, we have to redo it because we're not working with any gram-negative cocci in this course. Gram-negative bacilli, there's page 75. Okay, so next chart around that 74 to 77 area is this chart here, gram-positive. And so, we had a gram-positive bacilli. You look at some of the characteristics of the cell shape. So was it club shape? No, it didn't look like it. It was pretty uniform. Extensive branching, nope. And so this means that we're probably dealing with Bacillus or Clostridium genera. Okay, if it's gram-negative, we would go down to here, look at those, and see which fits best. And so we're going to deal with Bacillus and Clostridium bacilli. And so there is another one we're going to look at to narrow it down a little bit. We can look at this one. Is it spore forming? If it is yes, we'll do a positive. We'll go and consider Bacillus and Clostridium, which we kind of confirmed on that other chart, but this is a more detailed chart. And then we can quickly rule out one or the other. Bacillus is going to grow in oxygen presence, and Clostridium requires no oxygen. And in fact, it's toxic to us. Okay, and so that'll lead us to, again, probably to support the Bacillus. And then we got to go to the chart that is Bacillus species, because there are tons and tons of bacilli. Okay, and so then we'll do a starch hydrolysis, and we'll have explanations and show you these tests. Here's the group that it would be if it was a positive starch hydrolysis, this side if it's negative. If we do go positive, then we'll look at another test called Voges-Proskauer, which is VP test, and then we look at the positive negative. Then we can look at some of these other characteristics. Here's a citrate test, here's a sodium chloride test, and then we can narrow it down to basically a couple, in this case, if it went to B coagulans, then it would just be that. If two of them would be Bacillus subtilis and B licheniformis. And so then you basically get it down to one bacteria. And once you get it down to that final answer, you're hopefully supporting your hypothesis. So in order to have this be a usable result for a physician or for other healthcare people, you have to support your data with results, or support your hypothesis with results. And so, even if you suspect it might be Micrococcus luteus, based on a pigment color maybe, you still have to have the subsequent data before you can tell someone that it is, in fact, Micrococcus luteus. Okay, so you have to have your data has to support your hypothesis. Otherwise, your hypothesis has to go out the window and you have to come up with a new one that is supported by your data. And that's just simple science. But keep in mind, you want to get this right, because if this were in real life, you know, the physician or healthcare provider of a patient is relying on the identification of the bacteria in many cases to be able to treat it properly. And so this is just an introduction to unknown identification, and the people in the lab tech programs and lab techs, they do this day in and day out. They have some different protocols for patient uses depending on where the sample came from. If it was a sputum sample or a bowel sample, they would have different protocols because they're looking for, first off, you know, the normal bacteria that are going to infect that area. And then once they have, you know, the bacteria identified, they'll also look to see what antibiotic is effective against that. And they will put on antibiotics to try to grow the bacteria in the presence of it, and they'll look to see if the bacteria grows or not. If it doesn't grow, then that's good, because then it tells the physician what antibiotic would be useful for this infection. All right, that's it for the unknown. Ask questions when you have them. You know, Christy's online, and you can email me as well if you need questions. Thank you.